Everyone, welcome to our class. As we all know, we are currently studying the chapter Respiration and Excretion of class 9th textbook. Let's have a look at this image. Can you tell me which organ system is this? You all may have guessed already. It's the excretory system. Let's rewind what we have learned so far. Excretion is the process of removing waste products from our body. The major waste formed in our body is ammonia, which is extremely toxic and should be removed immediately from our body. Therefore, our body converts ammonia into less toxic urea. This urea is filtered in kidney and converted to urine and expelled through the urethra of urinary bladder. So now, let's look at how this urine is formed. Let's first observe this video and get an idea about how urine is formed.
The kidneys filter blood plasma, removing metabolic wastes, toxins, and excrete them in urine. During this process, they also maintain constant volume and composition of the blood, or homeostasis. Blood enters the kidney via the renal artery, which divides into smaller arteries, then arterioles. The arterioles get into contact with functional units of the kidney called the nephrons. This is where blood filtration and urine formation take place. The filtered blood then passes through a series of veins and exits the kidney via the renal vein. The urine is collected in collecting ducts and leaves the kidney via the ureters. Each kidney contains over 1 million nephrons. A nephron consists of two major parts, a glomerular capsule, or Bowman's capsule, and a long renal tubule. Renal tubules of several nephrons connect to a common collecting duct. There are 3 steps in the formation of urine: glomerular filtration, tubular reabsorption and secretion, and water conservation. Blood enters the Bowman's capsule via the afferent arteriole, passes through a ball of capillaries called the glomerulus, then leaves via the efferent arteriole. The afferent arteriole is significantly larger than the efferent arteriole, creating a blood flow with a large inlet and small outlet. As a result, the blood hydrostatic pressure in these capillaries is much higher than usual. Hydrostatic and osmotic pressures drive water and solutes from blood plasma through a filtration membrane into the capsular space of the nephron. The filtration membrane acts like a sieve, allowing only small molecules to pass through. These include water, inorganic ions, glucose, amino acids, and various metabolic wastes such as urea and creatinine, and make up the glomerular filtrate. The amount of filtrate produced per minute is called glomerular filtration rate, GFR. GFR is kept at a stable value by several feedback mechanisms within the kidneys, known as renal autoregulation. GFR is also under sympathetic and hormonal control. GFR regulation is generally achieved by constriction or dilation of the afferent arteriole, which causes the glomerular blood pressure to fall or rise, respectively. In a healthy person, the total filtrate volume amounts to between 150 and 180 liters a day. However, only about 1% of this is excreted as urine. The remaining 99% is reabsorbed back to the blood as the filtrate flows through the long renal tubule. This is possible because the efferent arteriole, after exiting the Bowman's capsule, branches out to form a network of so-called peritubular capillaries, which surround the renal tubule. The first part of the tubule, the proximal convoluted tubule, reabsorbs about 2/3 of the filtrate. In this process, water and solutes are driven through the epithelial cells that line the tubule into the extracellular space. They are then taken up by the peritubular capillaries. Sodium reabsorption is most important, as it creates osmotic pressure that drives water and electrical gradient that drives negatively charged ions. Sodium levels inside the epithelial cells are kept low, thanks to the sodium-potassium pumps that constantly pump sodium ions out into the extracellular space. This creates a concentration gradient that favors sodium diffusion from tubular fluid into the cells. Sodium is absorbed by symport proteins that also bind glucose and some other solutes. Nearly all glucose and amino acids are reabsorbed at this stage. About half of nitrogenous wastes also reabsorb back into the bloodstream. The kidneys reduce the blood levels of metabolic wastes to a safe amount, but do not completely eliminate them. Some of the reabsorption also occurs by the paracellular route through tight junctions between epithelial cells. At the same time, tubular secretion, where additional wastes and other solutes leave the bloodstream to join the tubular fluid, also takes place. The processes of reabsorption and secretion continue in the nephron loop, the loop of Henle, and the distal convoluted tubule. These parts of the tubule also have some other important functions. The main function of the loop of Henle is to create and maintain an osmolarity gradient in the medulla that enables the collecting ducts to concentrate urine at a later stage. The ascending limb of the loop actively pumps sodium out, making the medulla salty. The descending limb is permeable to water, but much less to sodium. As the water exits the tubule by osmosis, the filtrate becomes more and more concentrated as it reaches the bottom. The ascending limb, on the other hand, is permeable to ions, but not water. As a result, the filtrate loses sodium as it goes up and becomes more diluted at the top of the loop. The medulla is in equilibrium with the loop and hence has the same salinity gradient, saltier, at the bottom. Reabsorption and secretion in the distal convoluted tubule are under control of various hormones. This is how the kidneys respond to the body's needs and adjust the composition of urine accordingly. The collecting duct receives tubular fluid from several nephrons. The main function of the collecting duct is to concentrate urine and therefore conserve water. This is made possible by the osmolarity gradient generated by the loop of Henle. As it gets saltier deep in the medulla, the filtrate loses water as it flows down the collecting duct. The collecting duct is also under hormonal control, so it can adjust the amount of reabsorbed water according to the body's state of hydration. For example, when the body is dehydrated, more water is reabsorbed back to the blood, and the excreted urine is more concentrated.
By this video, I guess you all might have got an idea about how urine is formed. Let's discuss the process in detail. Urine formation includes 3 steps: ultrafiltration, reabsorption, and secretion. Let's look at each step one by one. Urine formation takes place in the nephron of kidney. The different parts of nephron assist in carrying out each steps in formation of urine. Step 1. Ultrafiltration. When the blood carrying waste reaches the glomerular of nephron, ultrafiltration occurs. Filtration occurs through the small pores of glomerulus and a residual fluid called glomerular filtrate is formed. Glomerular filtrate consists of water, glucose, amino acids, sodium, calcium ions, vitamins, urea, uric acid, creatinine, etc. Step 2. Reabsorption. The glomerular filtrate flows through renal tubules to the collecting duct. The essential components in glomerular filtrate is reabsorbed in the collecting duct. The rest goes into the blood. Step 3. Secretion. Excess components that are retained in the blood are secreted into renal tubule. From renal tubule, this fluid reaches the pelvis through collecting duct. This fluid is called urine. Urine reaches the urinary bladder through urethra and gets expelled out of the body. Urine consists of water, urea, sodium chloride, potassium chloride, calcium salts, phosphate, uric acid, creatinine, etc. By the completion of these steps, which occurs in the nephron of kidney, urine formation is done. Urine is highly concentrated and is removed through the urethra of urinary bladder. As we have now learned about the process of urine formation, let's do a small activity to check your understanding level. Let's try to identify the steps of urine formation from the statements given. The first statement is, in which step is glomerular filtrate formed? I hope you all have guessed the answer. It's ultrafiltration. Let's look at the next statement. What is the step that occurs in renal tubule? It's reabsorption. And the last statement, which step takes place after reabsorption? It's secretion. The answers can be seen on the screen. I hope everyone has answered them right. We have discussed the whole process of urine formation. Let's review our knowledge with some more questions to increase one's knowledge depth. The first question is, what are the 3 steps involved in urine formation? The answer is ultrafiltration, reabsorption and secretion. Next, where does the whole process of urine formation takes place? Answer, nephron of the kidney. Next, what is glomerular filtrate? Yes, it's the residual fluid after ultrafiltration. So far, I hope everyone is able to answer the questions. Let's look at the next question, shall we? Which is the second step of urine formation? It's reabsorption. Next, where does reabsorption takes place? It's in renal tubules. Let's look at the next question. What is the function of collecting duct? It collects urine. And last question, what is urine? Fluid formed after ultrafiltration, reabsorption and secretion and flown into the pelvis through collecting duct is called urine. From all the questions discussed, I hope today's topic, urine formation, is clear to everyone. Let's wind up the class after giving a follow-up activity. Draw the diagram of nephron and prepare a note on urine formation. With this, we shall end our today's class. I hope everything is clear. See you all next class. Thank you.